Hi, welcome to join me on this video where we will be focusing on the skeletal muscles. So earlier we reviewed also the cardiac and the smooth muscle, but the skeletal muscle will be our main focus here. Let's start by talking a little bit about the general anatomy of a skeletal muscle, and here we are going to look a bit larger view than just individual cells which you have been introduced to earlier. Did you know that approximately 42% of male's body mass is of skeletal muscle? Then what do you think that this number is for the females? Well, it turns out to be approximately 36%, and this difference can be explained at least in part by the presence of testosterone. It allows for the muscle fibers a greater ability to enlarge. So this is a starting point when you consider the role of anabolic steroids that some may take to grow bigger muscles with the help of hormone replacement therapy. But now we are going to approach the skeletal muscle as an organ. And this organ is made of different tissues, and these share some important features. In addition to the connective tissue that wraps around the muscle structures, we have nerve and blood supply to the muscle, and of course attachments to allow it to anchor to its origin and insertion. So each muscle is supplied by a nerve, artery, and a vein or veins. This is because muscles that are working hard require also a huge amount of oxygen and nutrients. In addition, the waste products need to be removed quickly and efficiently. Each skeletal muscle, as well as the muscle fibers, are covered in connective tissue. These support and reinforce the whole muscle, so there is a number of layers that cover these structures, and let's review these from most external to more internal. So, epimucin is dense, irregular connective tissue that surrounds the entire muscle. It may end up blending with fascia. Then perimucin, which is fibrous connective tissue that surrounds a group of muscle fibers known as fascicles. And finally, endomucin, this is fine areolar connective tissue that surrounds each muscle fiber. So these are the key connective tissue shells of skeletal muscle, and your textbook provides a nice review of these even in a table form. The other concept that I want to mention here relates to the fact that as muscles cross joints, in order to generate movement of that joint, they must attach to bones at different parts in proximity of that joint. So what we end up finding is that many anatomy and physiology textbooks are very big on talking about origins and insertions of muscles. Personally, strictly anatomically speaking, I'm not too concerned if you can differentiate between the two. What matters to me the most is that you know the endings of muscles that we cover, whether they are origins or insertions. But for the sake of being consistent with your textbook, let's note that an origin is typically classified as the fixed attachment point, while insertion moves with the contraction of a muscle. But really the reason why I felt strongly that I want to include this slide is the picture here, and what I hope to convey to you as the big message. So muscles work on levers to produce movement. And of course, in the case of our body, these levers are the bones. So let's have a look at this concept a little more. Here we have a man lifting a stone with a lever. And the lever will be the stake that they are using, and in our case, it would be the bone, as I said, right? What else do we have? We have the effort going in. And then we have the load. These all we are familiar with, I'm sure. And finally, one more time to highlight the pivot that we have. So this is in this diagram the smaller rock that the man uses to help with the lever. So how does this all apply to the muscles? Well, it all comes down to how far apart the origin and insertion are from each other. And this is what generates a mechanical advantage. And there is actually a field looking into this, something that I have been involved during my time working in the universities, and it is known as articulation biomechanics. Think of combining engineering and anatomy. Sounds like a dream come true to me. And of course we apply this same concept to various objects in our day-to-day life. Okay, I think that we are ready to look at the muscle microanatomy and something known as the sliding filament model. So, as we saw, skeletal muscle fibers were long cylinder cells that contain multiple nuclei. We will zoom in and study their structures. Within a sacromere, which was the membrane around the muscle fiber, we have something known as myofibrils. There are many of these. These are rod-like elements, and they form about 80% of a muscle volume. Now, let's look at an individual myofibril. Each myofibril contains numerous sacromeres. These are structures located between two Z discs. And we are going to pay attention to these structures in more detail, but at this point, I want to highlight that this is the explanation for the striations that you see in the skeletal muscles. So we get dark bands where we have H zone and light bands where we have the Z disc. That is pretty cool, huh? So there is a reason for all these things that you are learning with me. Okay, let's continue with this concept a little further. So here is a bunch of structures for us to observe, but I want us to focus on really on the blue and red lines that you see here. And this is an enlargement of these. Let's see what structures we can see there. So we see again our sacromere, and sacromere is the smallest contractile unit of a muscle fiber. So it is our functional unit of the muscle. And there are myofilaments, and there are two that I want us to focus on in particular. So the actin is the thinner filament type shown on blue, while myosin is the thicker one shown on red. And at this point, my students are usually like, right, I do not believe that these really exist. Well, I have something just for you. Here is an actual electron microscopy image of these structures. Note that the actin and myosin filaments. Pretty cool, isn't it? So you might ask me now, why is this all important? And that is a very good question. And the answer is that understanding this structure helps us to understand on a microscopic level how muscle functions. For a muscle to contract, these filaments slide past each other. So you can think of it almost like little legs of myosin filament that walk on actin. So when muscle contracts, which means that it will shorten, these filaments will slide so that more and more of these will overlap. And you can see this in these two images, with the one on the left representing a fully relaxed sacromere of a muscle fiber, while the one on the right is a fully contracted sacromere. Please note that their length does not change. It is just the amount of overlap that we are manipulating here. And this is what we know as the sliding filament model. And I think that I will try to provide you a very simplified video of this concept on the next activity on your learning module. But let's wrap up this chapter first. Next, I want to talk a little bit about what causes a muscle to contract. And this is a figure that you can find from your textbook. You can study it in more detail there, but I am going to give you a summarized story. So first, nerve impulse travels down the nerve cell and it reaches the end of the nerve cell, which is known as axon terminal. And here, chemical known as acetylcholine is released to the synaptic space and it causes a change in the flow of calcium in the muscle cell surface. If you're anything like my traditional face-to-face students, you will be asking now that it is all very good, but why do I need to know anything about the chemical changes? And my answer to that would be this following. And I'm going to warn you here that we have some pretty potentially upsetting pictures, so if you're uncomfortable with that, do not watch the next part. Rigor mortis refers to the stiffening of muscles after a few hours from the death, and it lasts for somewhere between 1 and 4 days typically. And the cause for this stiffening is the chemicals, in particular calcium, that cause muscle mass to become rigid. And if we look at the detail, due to the calcium, myosin heads become bound to actin, resulting in this constant state of these filaments being locked, if you wish. So it really looks like the body is frozen in place. And it starts usually with the small muscles and then typically spreads from the heads to toes direction. So really the term rigor mortis describes it very well, as when translated it stands for rigid death. This is where we will be ending our talk today. I thank you for your attention and ask you to study rest of the material in your learning module. I will see you soon again.